This protocol enabled us to solve the structure of human TRPC3, a member of an important but understudied family of ion channels. The main advantage of this technique is the high efficiency with which it can be applied to express and purify a variety of proteins for both structural and functional studies. This method can provide an efficient protein expression and purification system for use in studying protein biochemistry and biophysics, and can be applied to a wide variety of ion channels and other membrane proteins.
First, check the relative virus expression by viewing the GFP fluorescence of the virus in a sample of the virus-producing culture. In a baffled bottom Erlenmeyer culture flask of sufficient size, prepare a desirable volume of a HEK293 mammalian cell suspension culture at a concentration of 3.5 to 3.8 million cells per milliliter in the expression medium, supplemented with 1% sterile FBS. Add 8% by volume of prepared P2 virus stock solution and incubate in an orbital shaker at 37 degrees Celsius and 135 RPM. At 12 to 18 hours post-infection, add 10 millimolar sodium butyrate. Incubate at 30 degrees Celsius for the amount of time necessary for optimal protein expression. After this, centrifuge at 2,880 times gravity for 20 minutes to harvest the cells. Wash and resuspend the cells in approximately 100 milliliters of TBS per liter of cells harvested. Centrifuge again at 2,880.